Unit 3, genetics. This is our first day of our genetics unit. And so first thing we need to do is talk a little bit about chromosomes. And you have in your reading, there's no textbook section, but there are two things under the OpenStax column that you need to do. And one of them is about what's on a chromosome. And so here's a little overview, but make sure you go and do both of those things. So on this particular one, we're looking at the CFTR gene, which that gene codes for a protein that helps remove salts and other ions from the cell. And if it's non-functional or misfunctional, then people end up with cystic fibrosis. And so it is located on, so in this particular slide, you can see that it's located on chromosome 7. And so that's the first thing you would notice is that there's a little code here. And from the code, you can know exactly where your chromosome is located. So chromosome number 7 is where it's located. Q is basically the long arm of the chromosome. So you notice there's a short arm and then there's a long arm. And so the centromere is this little part in the center that actually, the centromere is this part in the center that is not really a structure, but it's more like a genetic code that then can bind to another one, because during DNA replication, when DNA makes a copy of itself, you'll have two of these that are identical because there's a copy and they connect at the centromere during cell division. And so we'll talk more about that tomorrow, but basically just a little vocab. So P arm is the short arm, centromere is where it connects to another chromosome. It often has a little bit of a funky shape there. And then Q arm is the long arm. So this says that this particular gene is located on chromosome 7 on the short arm at position 31. And so you would go down and you would find position 31, like you would just know where that is because you're amazing. Actually, it's region 3, band 1, and then subband 2. And so that's kind of how that works. So that's what that is. So basically every gene has one of these. So when you're doing your genetic research project, you'll need to know yours. Where is your gene located? Okay. We will, and you'll need to read to get more information on that. So that's not everything you need to know. So when you're looking at chromosomes, so here's chromosomes right here. This is a human karyotype, which means this is what genes humans have. And so if you can, you should know that we have 23 chromosomes. And we have 2 of each. We have one paternal and one maternal, meaning we have one that we got from dad and one that we got from mom, biological dad and mom, even if you don't know who they are. And so, so basically what we're looking at is your longest chromosome is chromosome 1. They are numbered typically by size. We have a little bit of a couple of them that are a little bit out of order, but they're way down here. And you don't need to know that, like which ones are out of order. But even though these look a little funky here, now you have to remember you're actually pulling DNA out here and karyotyping it. And so clearly they're not going to be like perfect textbook examples. Some of them are going to be wound really tight on their histones. And so you're going to see some differences. Even though they look a bit different, they still code for the same traits. And that is one word that I want you to know: homologous chromosomes. So homologous chromosomes are chromosomes that code for the same traits, but they are not identical. Okay. So like chromosome 1 here has 2, one from mom, one from dad. Those are homologous because they code for the same traits. So here's our chromosome 7 from our cystic fibrosis. Each of these has a CFTR gene on it. Now, hopefully they're both functional, non-mutated genes so that there's no chance that this individual would have cystic fibrosis. But on the Q arm, down at the bottom, and region 3, band 1, so about right there, on both of them, you have a CFTR gene. And so that means that they are coding for the same traits. They're coding for this. They have the same genes on them. Now, they might have a different version of the gene, what we call allele. They might have a different allele. So one of them might have the mutated form of CFTR, and the other one might have the functional one of CFTR. But regardless, they have the same genetic information on them. So we call them homologous chromosomes. They code for the same traits. Okay. And so when we get into genetics and we're talking about things like dominant and recessive, we're actually talking about the different alleles. So say this one on the right here has the CFTR mutation. where it would code for the bad, the misfunctional protein that's supposed to be a channel protein to get stuff out of the cell, but doesn't work properly. And say this one over here actually has the correct form. Okay, well, we would call that heterozygous because they are different. So hetero means different. And so these are heterozygous. Now the question is, what is dominant here? So if you have the CFTR gene that actually codes for a working protein, it works well enough that you don't show cystic fibrosis. And so it's okay for you to just have one. Okay. Then this one doesn't code for the protein, but it doesn't really do like the protein doesn't work if it's coding this CFTR, but you don't notice that if you have a working one over here. That means the mutated form of the CFTR is recessive. Why? Because you have to have 2 chromosomes coding for that in order to see the change. Otherwise, you're not going to see a difference in a typical individual because you have a working protein that's formed right here. And so you're good. Okay, so that's when we say dominant and recessive. Now, what would be dominant trait? A dominant trait would be if one copy of it screwed up the whole thing or just made the change. I guess it doesn't have to be screwed up. We have dominant traits that are definitely not screwed up, like the hanging down kind of earlobes, things like that. It's not like a mistake. It's just you only need 1 chromosome to code that way in order to have that feature. That's dominant. Okay, so it's recessive if you have to have 2. Now, if you have to have 2 to show the phenotype. Okay, so what's going on here? Let's see. Looks like we have a healthy, as far as we can see. Now, only thing we can see here is do we have 2 chromosomes for every, do we have both of them, both homologous chromosomes for each of the numbers? And yes, we do. We don't have any trisomies, meaning 3s. We don't have any missing ones. This is female because we have 2 X's here. So females actually have 23 homologous pairs. Males have 22 homologous pairs and then a non-homologous pair for their last one. We'll look at a male karyotype next. Here it is. So you can see they're all kind of quirky and weird looking. Okay, that's just because they've been manipulated and it's kind of a mess doing that. It's hard work to make a karyotype. But you can see here you have an X and a Y. And that X and that Y is the non-homologous pair that means that would express as a male. And so otherwise you have 2 of each. So this is a normal karyotype. Okay. Now I wanted to show you this just because it's kind of different. A lot of times you'll see these. Now you'll notice all of a sudden I have a bunch of X's where before we didn't have X's. So this is like a normal cell doing its normal function. This is right after they have replicated. So they've made copies of themselves. So this happens in the S phase of the cell cycle. Think back to your freshman bio days. We're going to go over that, not in this unit. We're not going to do cell cycle in this unit. We're just going to talk about meiosis. But in the S phase of the cell cycle, the DNA copies itself. And look, they're attached at the centromere right there. It's a centromere point. This is 1 chromosome here, or actually, that was a lie. This whole thing is 1 chromosome because this is a sister chromatid and that is its sister chromatid, and they are completely identical. There is no different genetic information there, so we don't consider it a different chromosome unless it's in its own cell and functioning. So when it's in an X like this, we actually would say that this whole X is 1 chromosome, okay? It's kind of a little nuance. But I just wanted you to know that if you see an X's like this, it just means that they're in the process of making copies of themselves and everything. And so it's this would just be for a short period of time, a karyotype would look like this. However, we tend to see them a lot. So I think it's just because they're a lot easier to see because they are like doubled in their DNA. And so it's a little bit easier to like figure out what the heck is going on. Male or female? Male. Typical, or do we have some differences? Typical. Okay, tomorrow's meiosis day. So I'm not going to go over every step of this. I just want you to know where the karyotype comes from. So here's your first cell, and it's doing its thing, and you can see their X's, so they've been replicated, and then they're pulled to each side. This is, you did this a little bit freshman year. We'll go over a little more detail in AP, and then they split, and then they split again, and you get these 4 cells. So this would be like 4 sperm, okay? And then one of these goes here and meets up with, like one of these would, if fertilization occurs, which it doesn't most of the time, but one of them would meet up with an egg. Let's say these are all sperm. And so let's say that's one of those. Now notice it only has one chromosome in it. That's not a human. Obviously, we would have 23. Meets up with an egg. Okay. And then they go together. And this is where the karyotype comes from. So like say this is chromosome 1 for humans. And behind it where you can't see, there's 22 more. I mean, they have to be in there. They're all in one cell. And so then this one is chromosome 1, and behind it is 22 other chromosomes. And you can only see this first one. Well, then this would be like the 2 chromosome ones in the karyotype for the offspring. Okay, so that's when I say one came from dad and one came from mom, and those are the chromosomes you got. Okay, we'll go over all the steps of meiosis tomorrow. And I do want you to make sure that you're keeping up with the vocabulary, because that's the hard part. Notice this haploid here. And this is a diploid, and this is a diploid, and that's a diploid, and that's a diploid. So you're going to need to know haploid from diploid for sure. We'll go over the vocab a lot more tomorrow. Okay, so then I want to look at this one, because what happened here, male or female, first thing you do. It's 2 X's, so it's female. And we have something weird. What do you see? You have a trisomy 21. This is Down syndrome. But I want to show you too that this is a pile of chromosomes and you have to sort them into their spots. That's why often they look a little wonky. So 3 right here, trisomy 21, 18, 19, 20, 21. This is Down syndrome, okay? Normal male right here, got it, XY, normal. Edwards syndrome, trisomy 18. Pateau syndrome, trisomy 13. Klinefelter's, you have too many sex chromosomes. And so those are just different kind. Now these, you cannot use these in your genetic presentation because they aren't inherited the same way that others are inherited, and there's a big chunk on inheritance. And so if you chose to do one of these, you would be missing a lot of points because there's no way to answer those inheritance points. Okay. All right. So that was a really quick rundown on kind of what we're going over today. Make sure you do your reading. And then tomorrow you can meet me on the Google Meet for meiosis, or you can come watch the video again. All right. I hope you have a great day. I'll see you soon.